scientist, right?
Yeah, he worked for U.S. Ameren.
Yeah. So he was a scientist. Apparently, maybe mental problems, and he had clearance to work with these type of things, and he was the one sending when they found out. He killed himself, so we don't know exactly what the story was. All right.
Mr. Eggman, is the endospore the whole cell, Paul, or just the genetic information in the middle?
Endospore is the whole thing right here. Okay, that's the endospore, and this is a protective layer around that. So, yeah, in this electron microscopy, that's the endospore. Here, this whole thing is the endospore. If I ask you what is this is the endospore. Okay? Okay, we said most of them are unicellular and very small. However, they stick together and form a variety of different shapes. We won't get into that. Staphylococcus, etc. There's a bunch of names for them. We'll leave it for the microcosm. But the things I want to focus is that even when you look at the prokaryotes, these are the, if you want to call it that, simplest form of cells, the very original cells. But they have evolved to become colonies, and that could be looked at as the beginning of becoming a multicellular, right? So you have groups that live together. There's a lot of research going in this area, finding more and more that forming colonies are very common in a variety of bacteria. It helps their survival, okay? And in some cases, even you had specialization in these cells. For instance, that's an example, a good example of this. This is a blue-green algae. The common name for it, cyanobacteria, is more scientific name. That's the genus. And Anabina, for this one, it is the species. Remember that? We're going to be using these. So that's the genus, that's the species. We don't have to remember the name of the species. Now, these are long chains of cells, okay, photosynthetic, so they use light photosynthesis. But they need nitrogen. Plants, things that do photosynthesis need nitrogen. We need nitrogen too. And they cannot use the nitrogen that is in the atmosphere, okay? N2 is not useful to them. So they have to convert it to some kind of a useful nitrogen, ammonia maybe, and so on. There are a couple of different ways that they do this. And so these cells have become specialized. Heterocysts is a specialized cell that takes nitrogen, N2 from the atmosphere, and converts it to a useful form, either nitrate or ammonium. Those are more useful. Does that make sense? So these guys do photosynthesis and feed this guy. This one is specialized and converts what? Converts nitrogen. What does your body do? Your body is pretty much specialized, right? Bunch of cells that are working together. Some digest food, some is your nervous system, some is muscle for movement, and so on. So, beginning of what? Again, multicellularity, if you will, right? From evolutionary point of view. All right, how do they reproduce? We've talked about this. Quick review for you. Asexually, so they have, we said, one nuclear piece of DNA, and that piece of DNA is linear or circular? Circular, right? And they have to make a copy of that. At some point you have two, and then the cell divides and breaks into two cells. This is called asexual reproduction, and more specific name for it is binary what? Fission. It breaks from the middle, you get two cells. This doesn't happen like this always in every group, every type. Some of them, they don't do binary fission, and they do it through with the DNA, and then they get bigger. All right, so there are variations. But the principle is the same. The DNA is copied, cell gets bigger, a portion breaks, and you get another cell. Okay, in addition to the chromosome, the one piece of circular DNA, you also know that they have plasmids. Different types of plasmids, and they carry specific genes on those plasmids, and they pass them to each other. Okay, now. So when we look at prokaryotes, in terms of their diversity and their success, maybe we can look at three major things. Rapid reproduction. Many of these, if they have the right conditions, food, good temperature, they can divide every 20 minutes and double their population. Okay? So it's a quick way of reproducing. When you reproduce a lot, also there is chance for what? A lot of mutation, because you're copying DNA all the time. Copying goes can have errors in it, and that's a mutation. So that mutation adds to what? Diversity, right? So that's how bacteria gets its diversity through, one way, through mutations. The other way that they get diversity, if you will, diverse genes, is through genetic recombination, all right? Which is another name for it, some folks will refer to as bacterial sex. We're combining what? Genetic material, even though they can reproduce asexually. So we're going to look at three different ways that diversity happens besides mutation: transformation, transduction, and conjugation. And you should know these words. The principle is easy. The words are a little harder to remember. Transformation, transduction, and conjugation. So let's take a look at it and see what this means. Transformation is when bacteria picks up DNA from its environment. So let's say this particular bacterial cell is sitting there floating, and another individual dies. And that cell breaks apart, the DNA is exposed. The guy who's alive can pick some of that DNA from its environment. Okay? Absorb it into the cell. And that, if it's a useful gene, will give him some new attribute. They can pass it to their offspring. They have become transformed. Remember one experiment we talked about? You had the rough type and the smooth type. They killed the one that was disease-causing, mixed them up, and the one that wasn't disease-causing became virulent. How did that happen? Picked up DNA through transformation. So that's a real thing. It happens in nature. Transduction. This is when genes are introduced with the help of viruses. Okay, viruses. Remember, they inject their DNA into bacteria, and then they repackage and make more viruses. So doing that, going from one type of bacteria to another, they may introduce new genes. And that's another way to get diversity. We call it what? Transduction. Alright? This diagram will show you how transduction works. So you have a bacterial cell, a phage injects its DNA into this cell. This cell has A plus gene, B plus gene, right? It destroys or chews up the DNA through enzyme, the DNA of the host, start packaging its own DNA, but once in a while what happens? This piece of DNA from here may be packaged along with their own DNA. And then when they infect the cell again, then they introduce a new gene to this bacteria. So this bacteria that is receiving the new piece of DNA has A minus and B minus type of gene, that's going to have also A plus. So it has a new gene, a new form of that gene, okay? Make sense? All right.
Question.
Yeah.
In the objectives, there was a question about discerning between generalized transduction and specialized transduction. I couldn't really find out the answer.
Don't worry about it.
Okay.
So cross those and just say they used to talk about it, but it's the same. So the amount of material will reduce. All right. Actually, they're working on reducing this book to a smaller version. There's already another book that has done it. Berkeley is using it, so everybody else is going to use it. If they can go with a smaller book, why should we, right? All right. Let's look at conjugation, but I can tell you later if you want. Conjugation, okay? In this case, what you have is two bacterial cells that are actually exchanging genetic material. So this is one, this is the other one, and they form this tubing we call sex pili, right, between the two. And now they can actually send DNA from one to another one. So we'll look at how that works. Right, sex pili, the tubing that connects two bacterial cells together. Through that, they give each other DNA. Larger view of it. Okay, this is what are some of the possibilities, some of the way that we've discovered these bacterial cells give each other DNA. So here you have an F plus cell, which means it has its own chromosome plus this plasmid. Plasmid is part of F plus. Okay. What happens if another bacteria shows up, they may form this conjugation, this tubing between them, and then the plasmid will make a copy of itself. And there are a bunch of stuff involved in the particular cell molecular biology. We'll go into detail. This is just giving you how does that happen, with all kinds of stuff that is involved. But DNA is copied and eventually travels through this pili to this cell. And what do we have now? Another cell that has a plus plasmid. Now this is huge. This is very important because this plasmid is carrying a bunch of genes. Okay? And now this bacteria has that also. Now if there was a gene here that would make this bacteria, for instance, resistant to penicillin, let's say, right? Then this one would become what? Also resistant to penicillin. If there was a gene here that helped you digest a particular sugar, this one now has it too. Right? So that's one situation. Another situation is a lot of times these plasmids may break into the chromosomal DNA and become part of that, like what they're showing here. So you have the chromosomal, this used to be a plasmid, now it's part of this. So we call these HFR cells, and they can also propagate, and so every time that they copy this, they also copy the other piece. But what they're showing you here, they can also give a piece of DNA to the F1 cell. Okay. How do they do that? Same thing. Piece of DNA is being copied, generally initiated by these plasmids, genes in the plasmids. As this is happening, okay, this guy got the plasmid, but what else?
F factor.
It also got another gene from this bacteria. See? A plus. part of the chromosome of this bacterial cell, but through the copying process, sometimes more genes are copied, and now A plus shows up in this particular bacteria too. Okay. So through conjugation, cells can be transformed, can be changed, and it's one way that they add to the diversity of the population. Are we together so far? All right, so spend some time with these two diagrams. There's a little bit of information there that you should know. All right, in terms of energy requirement, okay, and how they make a living, we can maybe look at this. Nicely organized in the book. So you should spend a little time with this diagram. There are several different ways that bacterial cells can make a living. So we categorize them in terms of nutrition. Number one, two large categories: heterotrophs or autotrophs. Autotrophs, they don't have to eat other living things for energy. Okay? Heterotrophs, they have to eat other things for energy and carbon. So let's take a look at different categories under here. Autotrophs, some are probably referred to as photoautotrophs. Others are referred to as chemoautotrophs. All right, so photo gives it away. Energy comes from where? Light, sun. Also, they need carbon, right? Why do we need carbon? To make all of those biological molecules, sugars, whatever, amino acids, etc. So the carbon for these guys comes from the atmosphere, CO2. So they're pretty much like plants, right? In terms of the nutrition. Sun is energy, CO2 comes from the atmosphere, photosynthesis. A bunch of algae, etc. that bacteria, cyanobacteria that I showed you. So, chemoautotrophs, these are very interesting. When they discovered this, it was kind of a surprise for scientists, because we thought that all living things were dependent on these photosynthetic organisms. Either they ate them or they have to use light. These guys turn out they don't need light, they don't eat other things, but how do they make a living? Sulfur. Chemistry. Their chemistry is very interesting and unique, different. They are sulfur-loving bacteria generally, okay, and they use sulfur to generate ATP. That's an interesting chemistry, and so they don't have to have light, but through chemical changes, they make ATP. Where do they get their carbon? From the atmosphere. So it's inorganic chemical. They don't need any living thing. They don't need light. They can survive with sulfur and other types of chemistry. So if I go back here, let's see. Okay, so we have photoautotrophs. These are some examples of those. We don't think about them a lot, but they're all over. They do photosynthesis. These guys produce more oxygen than trees on this planet, so they're really important to other living things. Okay? They live in lakes, oceans, and so on. When we look at heterotrophs, you can have photoheterotrophs. Photo says they use light, okay, just like these guys. But they get their carbon from organic material, these other living things. So they are dependent, that's why they call them hetero, on other living things, okay, for CO2. Chemoheterotrophs, organic compound for energy, okay? And organic compounds for carbon. So if we have to categorize ourselves in one of these, which one would us pick?
I don't have it.
Chemo, right? You eat meat and veggies and all that for energy and also for carbon. Yeah.
Are these the only modes of nutrition or just the major ones?
This is pretty much the fundamentals. There could be a little variations of each one, something slightly different, but they go under one of these categories. Okay. Some are parasitic, and you know they may not have any of these and use just the host. So those are different. But that could go under this one. Okay, depending how they use it. In terms of oxygen, to give you some ideas, they could be categorized in the following way. Obligated aerobes, these are the organisms that require oxygen. They have to have oxygen, just like me and you. We don't have oxygen, we die. Same with these guys. What do we need oxygen for again? To make ATP, oxygen picks up the electron, right? And the hydrogen at the end. Same situation for their basic chemistry. Others are obligated anaerobes, so oxygen is actually poison to them. Messes up their chemistry, okay? They use other things for electron transport. Could be sulfur, could be other molecules. So they go through fermentation or anaerobic respiration. Alright, they're slightly different, but same principle. Works without what? Oxygen. And then we have facultative anaerobes. These guys can do both, can survive if there is oxygen. If there is no oxygen, they switch. Okay, we call them what? Facultative anaerobes. Okay, remember this section, these several chapters, there's more things for you to memorize than actually connect. Yeah, the words are interesting, and we're going to ask you these words. So you should know these vocabularies, okay? So go. Learning some new vocabularies. Not as much processes, you know. We're not looking at their chemistry and all that. Okay, I already talked about this one. Again, living things require nitrogen, and the nitrogen fixing cells here, they do that, they take N2, get ammonia, and then it becomes useful to the rest of the cells to do photosynthesis. Okay. In terms of classification grouping, so when you look at bacteria, archaea, and eukarya, and we used to lump these together as one group, okay? We call them all Kingdom Monera, but today biologists are preparing to break them into two large categories, two domains here. So you have the eukarya, archaea, and bacteria. And which ones are more closely related to each other? Bacteria and archaea, or eukarya and archaea, or eukarya and bacteria? What? These two are more closely related, right? Very interesting. This is a surprise again to people who are doing this kind of work, because you would think these two are prokaryotes and they should be more closely related. But it turns out when they do look at molecular analysis that they have more in common with eukaryotes than with the bacteria. So the idea is that there must have been a common ancestor, whatever that might have been, that split and gave rise to the two branches, okay? When you deal with bacteria, as I said, by looking at them, you can't tell much, though you can tell the shape. But through DNA, RNA analysis, right,